So thank you everyone for this opportunity of talking about Roy, who I think for all the Cuban neuroscientists that of my generation was a very important and inspiring experience to be able to talk to him and discuss with him. And he laid the basis together with Professor Talia Harmony of the modern Cuban neuroscience school. So I want to be talking about Read the Mind. And the first thing we must remember is that in addition to the centenary of Evan Ray John's birth, we're approaching the 60th anniversary of the first article describing the P300 event-related potential, which was published in Science and which Roy co-authored. And the experiment essentially was presenting two stimuli. One would reliably predict the second, which could be a sound, a brief sound, or a brief visual stimulus. And in some conditions, that predictability would be broken, and there would be a uncertainty as to which stimulus was going to appear. And the basic finding, which you see here, this is 5 subjects, is that there was a positive wave about 300 milliseconds that increased when the stimulus arrived, and it was uncertain as to what was going to happen. In other words, the conditions that define the transmission of information. And this work, in contrast to previous work, focused on sensory that focused on sensory effects. This article highlighted the dependence of this event-related potential on stimulus uncertainty and information delivery and expectations. And we can say that this paper and other papers launched the field of cognitive neuroscience, especially cognitive electrophysiology. Roy also pioneered other experiments demonstrating the influence of top-down or endogenous factors such as mental set. And this is a paper he published in Acta Neurobiologica Experimentalis, and it's the response to an upright vertical stroke that in certain circumstances was interpreted as a number, and in others was interpreted as a letter. In other words, as 1 or as a letter I. And in several electrodes, Roy was able to demonstrate a statistically significant difference between the condition in which it was interpreted as a number and is interpreted as a letter. In other words, the same physical stimulus, according to the mental set, the interpretation produced different types of electrical activity. Now, the impact of the 1965 P300 paper is that it practically begat all cognitive electrophysiology. Up to date, there are more than 4,314 articles that directly cite that paper. But if you take into account all the papers, all the articles that are related with P300, this is an order of magnitude greater. And it's interesting to see that the P300 branched off that research into, I think, 4 basic directions. One is clinical applications, which is a pragmatic use by many authors, and that Roy incorporated into his neurometric methods. There's a very recent development in brain-computer interfaces, or large volume of work. And also, what I would call agnostic mental markers of processes, without trying to delve into what exactly are the mechanisms involved. And I should cite all this work in mental chronometry, and, for example, DeHane's use of this as a model of, and in his model of the global workspace, as to mark the moment information enters into the global workspace as opposed to specialized systems. However, despite much research, Roy personally invested less effort into the use of P300, in other words, to the basic understanding of the brain. I'll analyze why I think this was a wise move on his part. Why did Roy didn't invest more time into the use of P300 to try and do further basic understanding of brain function? Well, I think this can be responded very easily. A review by John Polich in 220 says, major questions, of course, are yet not answered after decades of research. What does the P300 mean? Where is it generated in the brain? Which of the cognitive operations it represents? So peer ERP research, which I think was a large bulk of the work in cognitive neuroscience, especially what was called psychophysiology, is what I call cognitive trap. It does not have the resources to really identify and respond questions like where's being generated in the brain, what does it mean, and what are the cognitive operations it does in it. And Roy recognized the need of avoiding this trap by formulating complex and multi-level theoretical hypotheses and testing them with diverse techniques that he pioneered. Intracranial recording of EEG with macro electrodes in animals, for example, intracranial unit and multi-unit recordings, metabolic mapping of neuronal activity in different mental states, or different, let's say, cognitive states with radio tracers, and the mathematical modeling of brain activity. So Roy really avoided this cognitive trap because he branched off and pioneered techniques that delved deeper into the function of the brain. I would like to say that this classical cognitive electrophysiology it had an important influence on an area which is, I think, very central to cognitive neuroscience, which is functional magnetic resonance imaging. So cognitive electrophysiology gave a theoretical toolkit of conceptual processing stages as an initial way of looking at brain activity. It gave it experimental paradigms and signal extraction methods like averaging. And this influenced the nascent field of functional magnetic resonance imaging. And you see in the first papers very clear this influence. However, what's not recognized is that functional magnetic resonance imaging had important reciprocal influence on classical cognitive electrophysiology. For example, the use of the general linear model to extract event-related responses, there are now toolboxes that apply the same methods that were developed for studying fMRI responses to ERPs, and which give cleaner responses and allow more complex modeling. For example, like multi-level hierarchical modeling of the event-related responses with what's called linear mixed effects. We now have toolboxes that allow us to handle this, and these were inspired by methods created by the fMRI community. More complete solutions to the problem of multiple testing. This is something that Roy and I remember Pedro Valdes discussed very early on, and how it was necessary to correct for multiple testing. For example, in an ERP, you have to test multiple electrodes and multiple time points, and using the Bonferroni solutions. But the most complete solutions to this came out of the theoretical developments, especially by Keith Worsley and Carl Fristron. And these techniques then returned to be applied to the cognitive electrophysiology. The application of multivariate pattern analysis or decoding techniques began in functional magnetic resonance imaging and is now being applied to ERP research. And the fitting of encoding models to describe neuronal function and testing deep neural networks against the fMRI data. So also all these areas are influencing cognitive electrophysiology. And of course, each time one of these aspects is tested in cognitive electrophysiology, it has an influence on the field of functional magnetic resonance imaging. Now, I think the most important thing to analyze is something that already Yuri has been talked about in his presentation, is that the theoretical landscape around the 1970 was heavily marked by the work, for example, in the visual system of Hubel and Wiesel's research, with their groundbreaking work that began in the late 50s and continued over 2 decades with several key findings. specialization of visual cortical neurons with their classification of simple, complex, hypercomplex neurons, ocular dominance columns, critical periods in visual development, and all this work was recognized in a Nobel Prize in 1981. And the idea of simple circuits that explained how more complex feature extraction, as they called it, could occur along the visual pathway. And the basic idea is that each neuron, let's say, visual neuron, had a selectivity, for example, to the orientation of edges and a position in space, which was invariant, and that allowed coding of information from the external world. And the very exciting findings were that for example, the center of the receptive field, the place it was, the eccentricity of it, had a linear relationship with the size of the receptive field, which mirrored many psychophysical and neuropsychological findings. So this led to a single cell emphasis on explanation of complex neural functions, the idea that single cell coding of stimulus underlies complex neural functions, that neuronal properties are relatively stable if connectivity is invariant. Everybody recognized the possibility of changes in connectivity would change the properties, and neural function driven basically by external stimuli, exogenous factors. And to recapitulate, it would be basically deterministic plus some noise and a high degree of localization. An example of an extreme of this idea is something that Roy discussed very much with his friend Jerzy Konarski. And as Roy reviewed, Konarski assumed that at a sufficiently high level within an analyzer system, unitary perceptions were mediated by the discharge of single neurons called Knox-like cells. And of course, this was dubbed at one moment as grandmother cells, cells that would code or represent someone's grandmother. And the work of Rodrigo Quiroga and collaborators effectively found highly selective neurons in the middle temporal lobe. For example, this is a neuron that responds basically to the face of an actor, Steve Carell, but not to other faces. And you see down here, this is the number of spikes. It's very selected for this group of photographs that represent the same face. Of course, there's also the Jennifer Aniston neurons. So all of this is part of a position where an emphasis is on single cell coding. Now, John's most groundbreaking work was this provocative championing of a different conception of brain function. The idea that mental activity corresponds to population activity, statistical not switchboard, highly distributed and redundant representation of information, that the role of neurons are flexible, that neural function is largely driven by endogenous factors, and is basically statistical in nature. And there's an important annual review of psychology in 1978 that summarizes many of these positions. And there was a similar work by Roy on unit activity with his colleagues Ramos and Schwartz, where they trained cats to respond to pedal A with a visual flicker at one frequency, say 1 hertz. They respond to a pedal B with a second frequency, let's say at 4 hertz, and then present ambiguous stimuli at an intermediate frequency, 2 hertz. And ask if the measure if it responds to pedal A or pedal B, if it interprets it as one stimulus or the other. And a movable microelectrode was implanted that permitted recording unit activities along a cortical thalamic trajectories. And this was very innovative for this moment. These techniques now have been developed tremendously, but at that moment, this was very innovative. And basically, he found that some cells were very stable to the same stimulus, independent of the interpretation made by the animal, the behavior response. But some cells, there was a variation in the post-stimulus histogram of the neuron firing that reflected if the animal selected one pedal or the other. In other words, that these visual neurons, this is visual cortex, were modulated by the interpretation. And 30% of their neurons in this paper showed this phenomenon. But how did Roy's ideas fare? For a time, these ideas were attacked or ignored. The consensus was slanted towards the single neuron doctrine. The technology for decades did not favor adequate testing of the ideas and precise Model formulations were difficult due to the limitations of computational resources. Recent years have seen a shift to a more nuanced position. We see some of this in Yuri's presentation. I want to mention this work. This is work of population receptive field mapping in humans using fMRI. So using wedges or rings of moving stimuli, it's possible to map the receptive field of a population of voxels. And this is essentially a human replica of Hubel and Weisel's work. And many of the same results are produced. For example, with eccentricity, the size of the receptive fields grows, and the slope of this relationship increases as we go forward in the visual pathway because receptive fields become larger and larger. Now, despite this being a replica of Hubel and Weisel's work, let's say at a population of neuron level, there's very interesting things to be said, for example, about PRF mapping when tasks changes. This is an article by Kendrick King and his collaborators where they mapped receptive fields using faces as stimuli when the subjects had to report a digit in the center of the field, of the visual stimulation field, or a dot that appeared on the faces, or they concentrated on identifying the face and comparing to a template face, one back task. So though the stimulation was the same, the task different. And the important finding we can say here is that PRF properties vary with task, and that both the eccentricity of the population receptive fields, the size, and the gain changed significantly when the subject was looking at the faces compared when he was doing a digitet or a dot task. In other words, not processing actively the face structure. So this is interesting because it shows even when we do Hubel and Weisel type receptive field mapping in humans, the properties of the receptive fields are very plastic. They change with the task, and this means that there's a remapping of the property of these groups of neurons. And Kian Quiroga and his colleagues have reached the conclusion that these neurons he described in the middle temporal lobe are not grandmother cells. They're sparse coding because it's implausible there's only one, and this is from a paper they wrote in Transit Cognitive Neuroscience. It's implausible there's only one cell responding to a person or a concept, because the probability of finding this in a cell out of 100 million neurons in this area would be very small. From the number of response units, they've argued by Bayesian probabilistic arguments that it's possible to estimate approximately out of approximately 1 billion middle temporal neurons, fewer than 1 million represent a given percept. So this is very far away from the Gnostic units that were being discussed decades ago, and that some units fired to more than one individual in an invariant manner. So the other thing is that now we have new tools, for example, like calcium imaging, and there's studies that permit the exploration of population neurocodes and show that when you work you examine the multivariate coding of stimulus, this is more robust to the variability and the noise than when you analyze activity only from one neuron. And though detailed modeling of Roy's approach was not available, his ideas, to test his ideas, this is now potentially possible with large-scale network simulation. Here I'm showing one of the early models. This was practically just a few years before Roy passed. This is the H-Max model developed by Serre and Poggio and Oliva. And they developed a model that initially was relatively simple, trying to model the properties of simple cells, complex cells. But now this has evolved into, we know now there are deep neural networks which contain hundreds of thousands of neurons and a very complex internal connectivity. And though some of these architectures do not really resemble how the brain works, there's a great deal of flexibility, very new models. And this means that a language has been developed which can be used to model this activity. And these simulations reconcile the mass action of units and their specialization. So we have mass action, but specialization. So to conclude, Erwin Roy John was one of the parents of cognitive neuroscience. He made groundbreaking contribution in experimental techniques and methods of analysis, including the first applications of multivariate statistics to neurophysiological data. He championed population-based statistical models of neural basis of mental activity, raising concerns that resonate today. These models initially could be tentatively formulated and tested, but the newer techniques like a high-field fMRI, calcium imaging, to mention two examples only, now offer new data that allow better testing, and advances in computational neuroscience and neural network modeling now allow more precise formulation of these models, and a synthesis will probably emerge, reconciling the single-cell data and the population models with much more complex theoretical positions. And I just want to finish this presentation showing this cat computer that was a donation that Roy made and a group of his colleagues. In a moment, we're still unable to receive equipment from the US, but a lot of colleagues pitched in, and they bought this very first primitive computer, and we were able to record in Cuba our first ERPs with this system. And they had a plaque that said, "To the Cuban people from their American friends." And this is something we felt very much. And I would like to finish with this photograph of Roy. We're sitting near the National Center for Scientific Research. There's Roy, there's Guido, our two Chilean colleagues, Holger and Ruturba, Talia Harmony, that's her back there, and that's me. And this is how I remember Roy, discussing always with a terrible ferment of ideas, generating a lot of critical thought and discussing among friends these ideas to make science advance. So thank you.